So thank you again for joining us today. We have three speakers joining us to talk about ancient DNA research. And again, I just want to mention before we start that we find ourselves in the horrible situation of the still ongoing conflict in Sudan. So our thoughts go out to the people of Sudan. And as sadly per the last 2 weeks, we hope for a peaceful conclusion and end to this conflict immediately. So jumping right in today, the first speaker that is going to talk to us is Dr. Kendra Sirak. Dr. Kendra Sirak is a research associate in the Department of Human Evolutionary Biology and Department of Genetics at Harvard University. Dr. Sirak's research focuses around the use of DNA methods to assess past human interactions, as well as developing novel protocols to advance the capacities of ancient DNA research. Dr. Sirak is currently involved with numerous global projects utilizing DNA research, among them several in Sudan, in particular with a focus on Qulub Nardi, where a high rate of DNA sampling success was achieved. So thank you for joining us today, and the floor is yours.
Okay. You can see my screen okay? It looks good? Yes? Good. Okay, great. Well, then here I go. Yes, thanks, Robert, for the introduction. I'm really excited to talk about this today. Anytime I could talk about Qulub Nardi is a good one. So I'm going to begin my talk today with a brief introduction to ancient DNA, because I know that this is a topic that not every archaeologist is inherently familiar with. So I think it's always good to start with a little bit of a crash course in this. So just basically, what is ancient DNA? What it is simply is DNA that's extracted from the remains of an organism that lived hundreds to hundreds of thousands, or now over 1 million years ago. We usually study ancient DNA from biological material, such as bones, teeth, or hair, but increasingly, we're studying sediment or cultural artifacts and recovering DNA from those substrates. You might have seen just a couple weeks ago, there was a very big paper about a DNA recovered from a deer pendant that was worn around an ancient Eurasian woman's neck. So I like to think about the study of ancient DNA as moving from bones to moving to base pairs. So how do we do this? The way that we do this is we take a bone or any sort of biological material, we extract the DNA from that using a watery substance that breaks down the bone cells or whatever kind of cells of the material that we're using is, and releases the DNA into solution. We prepare the DNA molecules to be read by a sequencing machine by adding small oligonucleotides to the ends of those molecules. Then we carry out sequencing, usually next generation sequencing, and we get this long strand of A's, T's, C's, and G's, which is our genetic code that we analyze. So more important than how do we study DNA, what is exactly the part of DNA that we want to study? And the part that we study as ancient DNA researchers are called single nucleotide polymorphisms. So breaking this down, single, one, nucleotide is one of the A's, T's, C's, and G's in your genome, and polymorphisms means many forms. So basically, at this single position, there are two or more options for the nucleotide. So this is why we call them SNPs. Now, this is the most common type of genetic variation among people, and every genome has 3 or more million SNPs that we can study, despite the fact that we're all 99.9% genetically similar. Now, when we study SNPs, we want to study them across the entire genome. So a human genome is 3.2 billion base pairs long. And what we want to do is study those polymorphic positions across the entirety of the genome. Now, this is a really powerful approach, and we call this a genome-wide approach. And the reason that we want to adopt this approach in particular is because by studying the genome of a single person, we're actually able to look at genetic information from hundreds to even thousands of that person's ancestors. And that's because a person has many lineal ancestors who pass down their genetic information to create the genome of that single individual. So you can see here, we have our individual, we have mom and dad, we have 4 grandparents, 8 great-grandparents, etc. And only around 20-some generations back, we have up to 1 million ancestors. Now, all of these ancestors contribute small parts of their genome to form the genome of the individual that we're studying. So this is a really powerful approach to understanding past genetic landscapes. So why do we study ancient DNA? We study ancient DNA to learn about people who lived at a specific time and known place in the past. And this is because humans have just not descended in simple ways from populations that lived at the same locations in the very distant past. Instead, we know that human history is characterized by extensive movements and mixtures and adaptations throughout our history. So what's been done so far? Just earlier this year, we passed the threshold of having over 10,000 ancient genomes published. So this is genome-wide data from 10,000 ancient individuals. And you could see this graphic here, which shows the distribution, and it shows the rapid growth of this field. However, one thing that you'll notice is that Africa constitutes only 3% of the entirety of these 10,000 genomes. So this is very little data. So why? Why do we have such little data from Africa? It's such a big continent. It has the most genetic diversity of any place in the world. It has a deep history of occupation. Why do we have such little data? The answer can be found in this map here, where you'll see that Africa is colored primarily red and primarily blue. And what these colors indicate is hot and dry and hot and wet. And the number one factor that contributes to the degradation of DNA is heat. So it is very difficult to get ancient DNA data from Africa. So the fact that we have so little is not for lack of trying. Instead, it's because when DNA is exposed to heat in an organism that is no longer undertaking repair mechanisms, so when an organism dies, the repair mechanisms that will work to repair fragments of DNA are no longer functioning. So DNA fragments and collects chemical modifications, and it becomes increasingly contaminated. So here you can see in a living person, we have these nice long strands of DNA with these little mutations, these SNPs that we want to study. However, in archaeological contexts, you could see that these white strands of DNA, which are the pieces that we want to study, are all mixed in with environmental contamination and are very short and very fragmented. And this makes the study of ancient DNA very difficult. However, there are things that we can do to mitigate this. Now, one thing we do to study DNA from Africa is we focus on one specific skeletal element. And this is the petrous part of the temporal bone, and specifically the osseous labyrinth, which is inside this petrous pyramid. Now, we know from previous research that this is an extremely DNA-rich part of the skeleton, and this is because of unique developmental and histological properties. However, we want to use this skeletal element whenever possible for ancient DNA analysis. So if you've ever worked with me before, you'll know that that is the element that I focus on most specifically in all of my work, and that has enabled our success for me personally working in Nubian contexts, and for others personally working in African contexts or other tropical or desert climates. So moving into the heart of this talk, I'm going to talk a little bit more about the genome-wide ancient DNA data that I recovered from individuals buried at the Christian period site of Kulubnardi. So before I begin, I want to acknowledge the ancient people that we're studying. They have been part of this amazing research program that has spanned over half of a century, as well as 4 of the advisors who helped me so much with this work, and entire teams of people who contributed a lot to this project. So probably don't need to say much of this to this group, but Nubia is this area along the Nile River from the first cataract at Aswan through the 6th cataract near Khartoum. And the reason that I'm interested in studying this as an ancient DNA researcher is that for many millennia, this Nile corridor has been a place of movement and exchange of culture, of goods, of genes between sub-Saharan Africa, Egypt, and further afield into West Eurasia. So this makes it a very exciting place to explore from a genetic perspective, both in the present and in the past. So In 1979, two cemeteries less than 1 kilometer apart were excavated at Kulub Nardi. One was on the island of Kulub, which is the S cemetery, we'll call it, and the other was on the adjacent West Bank, and we'll call that one the R cemetery. Now, archaeological and bioarchaeological analyses of the skeletal remains suggested that Kulub Nardi was home to a biologically related and culturally homogeneous population divided into two socially distinct cemetery communities. So what does that actually mean? So biologically related, researchers found no significant differences using discrete dental traits, cranial non-metric traits, or craniometric traits. And this is research that was done starting in the 1980s and expanding all the way through 2020 here. Now, culturally homogeneous. There was no difference in grave type, grave orientation, head or body positioning, and an overall lack of grave goods in both cemeteries, as would be consistent with Christian-style burials. Furthermore, there were no isotopically measurable differences in diets, so it looks like the people who were buried in each cemetery were eating roughly the same type of diet. Now, this idea of socially distinct. This comes from examination of patterns of growth, of stress, of disease, and age at death. So we could see that in the S cemetery community, there was more severe chronic stress and higher markers of this generalized stress, such as through cerebral orbitalia, and a much younger average age at death of only 10.6 years old, as opposed to about 19 in the R cemetery. So it looks like these groups had a very different lived experience, and that's what we wanted to study from a genetic point of view. So in a 1999 monograph on Kulub Nardi, Adams et al. Write, the combination of cultural and biological evidence suggests a wholly unexpected possibility that this region in early Christian times was home to two biologically and culturally indistinguishable but socially distinct communities, one of which was considerably better off than the other. And he also writes, for the moment, there is no obvious explanation for this anomaly. And that's where my research came in. Now, one of my big research aims was to begin an investigation of the genetic landscape of the Nile Valley before the spread of Islam in the late first millennium CE, which we know created a lot of biological and a lot of cultural changes. And then the second goal of my work was to address this question about the relationships among the people buried in the two cemeteries at Kulub Nardi who show these differences that are suggestive of a socially different experience. And we want to explore these using genomic data. So we screened 111 individuals from Kulub Nardi for authentic ancient DNA. And we enriched promising libraries, which showed evidence of having some DNA preserved, for sequences overlapping 1.24 million of these SNPs from all across the genome. And we obtained genome-wide data for 66 individuals, which is remarkably high for this context, but probably has at least something to do with the relatively recent time period that these individuals lived during. Now, we performed direct radiocarbon dating on 29 of the individuals, and were able to determine that the cemeteries were in use contemporaneously from about 650 to about 1000 CE. So I'll discuss three of the key results and tell you a little bit about how we got there. So the first result was that all individuals from Kulub Nardi were mixed with varying amounts of Nilotic-related and West Eurasian-related ancestry. So to explain what I mean by West Eurasian-related ancestry here, this is not necessarily saying that this ancestry originated in West Eurasia, but it definitely originated north of sub-Saharan Africa. So we use this term as kind of a catch-all to differentiate between sub-Saharan African ancestry and ancestry that is probably either North African or West Eurasian in origin. So we use principal component analysis, or PCA, to illustrate how the individuals from the two Kulub Nardi cemeteries relate to present-day people as well as to each other. So you can see the present day people here are represented in symbols of lighter color, and they're arranged along two clines that share a terminus right at the bottom right-hand side of this plot. So we want to focus specifically on this cline here that spans both of our PCs and spans from West Eurasian people, both ancient and present day, to people who spoke Nilotic languages and lived in Sudan and Ethiopia. And across here, we see this cline of people from Nubia, Sudanese Arabs, and Ethiopian people who speak Afroasiatic languages of the Cushitic and Semitic branches that are distributed along this cline. And the Kulubnardi Nubians project right over this cline, kind of spread, moderately spread across the cline. We see a couple of outliers here. So this gives us some suggestion that they are mixed between some sort of West Eurasian ancestry and some sort of African, sub-Saharan African, and probably Nilotic related ancestry. We are able to test this using a formal statistic called an F3 statistic, and we can confirm that the allele frequencies of the Kulubnardi Nubians are between Nilotic populations and West Eurasian populations. So a main question here is, where did this ancestry come from, this West Eurasian related ancestry? And what we want to do is to try to model the Kulubnardi Nubians as descending from two-way mixtures between a Nilotic sub-Saharan African related source and a West Eurasian related source. So we test all these West Eurasian related sources that you see here on the left, and we find that only one fits into this model, and that is from Egypt. And we find that the Kulubnardi Nubians can be modeled as having about 60% of their ancestry coming from an Egyptian-related source. However, Egypt is in Africa, so this doesn't give us an ultimate source of this ancestry. So we remove Egypt from the model and we run it, and we find then that the ancestry in the Kulubnardi Nubians that is West Eurasian related appears to derive from the Levant, specifically from the Bronze and Iron Age Levant. And we know archaeologically, historically, that there are deep connections between the Levant and the Nile Valley for many thousands of years. And we find that about 57.5% of the ancestry in the Kulubnardi Nubians can be modeled as being derived from this ancient Levantine population. Now, we find in response to that second question, that there's no evidence that people buried in the S cemetery were genetically different from people buried in the R cemetery. One way that we found this out was by looking at families. So 33 individuals from Kulubnardi had at least 1 and up to 5 genetic relatives in our data set. So this is first, second, or third degree relatives. And they shared 28 pairwise genetic relationships, forming 8 extended families. Now, the important thing is that for 7 pairs of relatives, 1 individual was buried in the R cemetery and the other was buried in the S cemetery. So this is consistent with a scenario of some fluidity between cemetery groups and doesn't provide any support for some sort of caste-like social system of division at Kulubnardi. We also tested this difference for genetic difference between the S and R cemeteries using a lot of statistical methods. So I'm not going to read them all over here, but we did do tests to test for statistically differences in allele frequency between these 2 cemetery groups, and we did not find any. Now, the third important result is that West Eurasian related ancestry at Kulubnardi was disproportionately associated with female ancestors. And I think this one is really interesting here. Because females carry 2/3 of the X chromosome in a population as they are XX, and males carry only 1 X, they're XY. Females also carry only half of the autosomes. So 2/3 of the X chromosomes, but only half of the autosomes. And this can be used to detect a signal of asymmetrical admixture between males and females. So this figure here is from Skoglund et al. 2016, and it's well described in that paper. So we use our QPADM models to compute ancestry proportions on the autosomes and the X chromosomes of the Kulubnardi Nubians. And we find that the West Eurasian-related ancestry, which we model using that ancient Levantine population, accounts for around 57.5% of the ancestry on the autosomes, but around 64.4% here on the X chromosomes. And this reveals that the West Eurasian-related ancestry at Kulubnardi was disproportionately derived from female ancestors. So overall, this highlights the importance of female mobility and suggests that it's possible that Kulubnardi could have been a patrilocal society, which fits with some of the oral history of people who live in this region. So to sum things up, this research is accessible. It is published open access. The paper can be downloaded on our website, and all of the genotype data are available. So you can download the raw files and analyze them yourself too, if you would like to explore more questions. And this is the accession number for those data. So thank you all for listening. And just wrapping up, I just want to take a moment and think about all of our colleagues who are facing this scenario of war right now. And our thoughts are with them all as we talk about their history today. Thanks.
Wonderful. Thanks, Kendra. That was a great presentation. Thanks for leading the way today. We will move right on into our second presentation, which will be provided to us today by Dr. Abagail Breidenstein. Dr. Abagail Breidenstein is a lecturer in the Department of Anthropology at Binghamton University. As a bioarchaeologist with a focus in the Nile Valley, a key aspect of Dr. Breidenstein's research focuses on assessing DNA from ancient contexts, having conducted research as part of the Paleogenomics and Mummies Studies Group at the Institute of Evolutionary Medicine at the University of Zurich, and currently as the Associate Director of the Nuri Archaeological Expedition in Sudan. We're so glad to have you joining us here today. Please, the floor is yours, Abby. Thanks.
All right, I'm going to skip through a lot of what I started because Kendra did such a wonderful job of introducing some of the hardships, but also some of the very important reasons of why we work here. So thank you, Robert, for the introduction, and as well for allowing me to be here to talk about my work. Because we all have a little bit something different to contribute. It's a constant theme within the teaching that I do, that this is a borrowed science and we're constantly re-upping our technology. And this is a very fast-paced field, and we need seminars like this to keep everybody in the know, to think about how to optimize this kind of research and understand this very, very important part of African history. So like Kendra said, I'm not going to spend too much time on why it's very important to talk about the Nile corridor, because thinking about how people left Africa is a huge part of why we look and use paleogenetics, but sometimes focusing our work inside Africa and what happened around Africa is something that paleogenomics is now starting to do. And it certainly has something to share about Africa's history, as well as what happened with migrations, what were the forces and the ebbs and flows of people for the continent that has been the most occupied and has the most genetic diversity of anywhere. So the Nile River itself is a corridor. It has been studied for centuries. Paleogenomics is in this position now to think about how to add ourselves, not as a means to replace the works before us, but how do we add to the vast array of evidence, whether it's archaeological, historical, ethnocentric, or some sort of cultural data to understand the Nile Valley corridor as a very important and significant place in history. Populations have been around here for tens of thousands of years in many different forms. And all of those, the rise and fall of many of these kingdoms is left on the landscape and has been studied for centuries. And most importantly, what people and my colleagues like us have been thinking about the most are all of those molecules that are trapped within the skeletons that are interred in these cemeteries, because they have these cultural practices of burying their dead and leaving it there, that we can keep learning from these individuals. So I'm going to speed through this since everybody here is an expert, that we're centering ourselves around ancient Nubia, and all of these factors and contextual elements need to be brought into the argument of understanding genetic data, because it is not in a vacuum, and thinking about these overlapping places. And this is just in the Nile Valley of Egypt, and ancient Sudan. It has not, this does not even include some of the very the vast kingdoms in Ethiopia, in West Africa, and sub-Saharan Africa. So this is even just a very tiny scope, but genetic evidence should not be seen in a vacuum and should be a supplement to the work that has already been done and continues to be done to understand African history. So genetic work itself is this window to the past to understand human variation in real time. We literally have a time machine when it comes to genetic past, and that's what is so fascinating about this this field. So this is an somewhat outdated, the same Caraway, the same guy from the magazine that put these out, that there is this bias in Europe, whether it is because there was so many other things in the beginning of genetics, paleogenetics, that it was easier to work with other materials than African materials. And there were a lot of mitigating factors when it came to issues with using materials from Africa. And as Kendra introduced, there are many more challenges when it comes to using materials from African contexts because of the environmental degradation. There's also these other factors of funding, somewhat of a Eurocentric bias, because there are many of the labs are in Europe, and that adds to some sort of a bias that's there. So just to highlight, the first one was published from an Ethiopian individual in 2014, I think, but I think the paper got a revision, so it's in 2015. But since then, it has exploded when it comes to understanding African history from a paleogenomic standpoint. So this is a map that I've been continuously adding on, and it still needs to be updated because I'm constantly trying to add things to this map, but understanding what African history was from a genetic standpoint used to just be a few few genomes here and there. But since then, we have 300 plus, but it's spanning 15,000 years of genetic history. And this is just a drop in the bucket when it comes to African history, and thinking of how many individuals are there. That's something like 300 plus, or moving towards 500,000 individuals from an entire continent that has been occupied for tens of thousands, hundreds of thousands, millions of years. That's really tough because it's just really a drop in the bucket. So for a really, really long time, this map looked different because there wasn't this columnarity item, that there was this huge dearth of genetic data from Sudan, from ancient Sudan. And so now that Kendra has published the Kolobnardi individuals, you finally start to fill out some of these places in Africa, but there is a huge dearth of them throughout this swath of the Sahel because of degradation. But all the way spanning from the Canary Islands to South Africa, there is a lot to be known from the paleogenomic standpoint. So now that we do have, and there should be one more that should be added that was published in December from the Kerma period should be added to this map as well. So these research questions that I'm mostly focused on for my dissertation work, and continuously I'm thinking about, a lot of it had to be, does it work? does it actually work because we have all of these mitigating factors that keep the DNA from us because it's already completely degraded. And what characterizes Nubian ancestry? What does it mean to have ancient Sudanese ancestry? Again, as Kendra introduced, the amount of people and that have been moving through this space for tens of thousands of years means it's going to be a very complex picture. Does paleogenomics have a place to help detangle some of that? And how can we marry it with archaeological, historical, and cultural data as well, and then layer on linguistics as well. Especially questions that paleogenomics can look at is, does genetic, your characterization of geneticness, or your what characterizes as genetic ancestry, does it change with cultural transitions? Do we have people that are moving in that may be from out of town, or are these people, especially during these the rise and changing of all of these kingdoms, are these people moving away, or are they staying with the regime change and adapting for sure? And how do we marry all of these lines of evidence when it comes to paleogenomic data? How do they come together to nuance these ideas or add to them? But in all, it's going to be always a very complex picture. So I think I wanted to focus on for this the work that I did, because I work in an area near the S bend of the Nile, and the preservation is even worse than you can expect. And so thinking about the optimization of ancient DNA, what are the things that we should be taking away? What should we be sampling, and how should we be sampling it, whether it's a cost-effective way or the least minimal, if paleogenomic is the kind of data that we're searching for, and that we have as a part of a project or are important to the stakeholders of these research questions. And so a lot of my dissertation work was the method optimization of working with 6 different populations from around anywhere from the 3rd to the 5th cataract, where variation in the environment and the deposition of the individual was different. And so getting this information out there is really important, and we do plan to publish in the next month or 2. So I'll just take you into the lab with me and show you all the things that I did, so that either they don't need to be repeated, or there are questions, specifics about it for understanding what should be done, what shouldn't be done, or what worked, most importantly, right? So the materials that I had were given to me as a steward in collaboration with many of the people that are here today. And the generosity to support my dissertation work was just astounding. And I was so pleased that I was able to bring around so many different populations, whether it was in space or time, as well as depositional environments. So I had I have about 240 samples representing 89 individuals from 8 archaeological sites in the Upper Nubia region. As the publication of the Petrus portion as being the gold standard, and it has more endogenous DNA, I selected for individuals that had good preservation of the Petrus portion, as well as teeth, for sampling to recover ancient DNA. 43 individuals of this entire cohort were trialed, and all of these individuals date from circa the Napatan period. We're not really sure when that individual was. A lot of these burial contexts need a little bit more work, or the radiocarbon dating failed, which is never a good sign. But all the way up until the Christian kingdoms, definitely late Christian kingdoms, especially through the El Kuru individuals. And so we have, as you can see here on the map, these are all the populations that I was able to bring individuals from and try out samples, all the way up in the 3rd cataract near Kiwaka. So these were individuals, just a few that had been a part of I think it was a rescue excavation, or just a quick trial to see how things were, as well as those that were generously given over by Tayyab. So El Tayyab through Tangazi, Deti, and Zuma. And so those are going to have a different depositional environment because these are very humid environments. These individuals were deposited in these somewhat catacomb-like burials. And so that was a very different environment. We had Getzali, which are far away from the river, very good preservation where nails and hair are still preserved. I specifically excavated the individuals at El Kuru, also a little too close to the Nile, close to irrigation, and close to modern agricultural lands. So that represented another place where if you have a cemetery in that situation, what do you do with it? And then there were individuals from the Berber Meroitic Cemetery, as well as TARP is the Tinga, was an archaeological rescue. That one was a rescue because they were putting in a plastics plant. And these were beautifully excavated and preserved from this rescue. So that gives you a little bit of context of where we are, of where these individuals came from. So I processed temporal bones where it was possible, or tooth samples to compare and contrast what was working better. So for tooth samples to be processed, first, any of the calculus was removed and saved. We have an external cleaning and a UV decontamination, so kind of killing anything that would have been on the outside. So that is all about, when working with ancient DNA, it is all about handling and making sure that the contamination is under control. So that means ablating the surface on the outside, so removing that external surface. But sometimes we need to make sure that we're not being overly eager with that ablation, because the cementum, this outside tissue on the outside of the dentin on the root, is actually very valuable for aDNA. So often we are sectioning or treating these teeth in various different ways, but collecting all of the tissue, even though it may look like it's not necessary. So we keep the calculus, we keep the cementum. And then for these individuals, I believe this is a tooth from Getzali. So that the roots are removed by cutting just underneath the cemento enamel junction, the CEJ that goes around the neck of the tooth, in order to have access, gain access to that very sterile dentine underneath the crown. And in many instances, I also crush the roots after ablating them and removing that cementum, just to again see where are the richest parts of the teeth for these individuals that are from the Upper Nubia region near this S-bend, because contamination and degradation is a serious issue. For temporal bones, this is a more invasive selection process of getting those tissue, but it is either drilling, hand drilling, or crushing that is done. So for these individuals, we are looking at the petrosal portion of the bone that is specifically here. And with what Kendra mentioned, this bony labyrinth is here, where it's reinforced with all of these really dense portions of hard cortical bone, I think it's cortical bone. And what you do is you cut right underneath on one side of it, again, to gain access to this sterile part of the bony labyrinth. But as you can see, many of these look really different. We have biological variation. Everybody's temporals look a little bit different. In fact, they're actually so unique that they should be CT scanned in order to look at familiar lines of understanding them as well. So these formations these morphologies of the temporal bone are so unique, they should be preserved before this invasive cutting. So for one method I also used was isolating this bony labyrinth by cutting away and hand shaping, or just kind of hand ablating this area here and crushing it in a sterile mortar and pestle called the bone crusher. That is just a system that I can use to quickly process and sterilely process individuals to make sure that I'm getting a lot of powder that is derived from the individual. For drilling of petrosal bones, it's the most sterile sampling method to date. So what I did for my dissertation, the drilling and the crushing were the most important. I did try some cryomilling. It really didn't work. I would not suggest anyone to use it because it's not helpful. There's too much contamination. It's too difficult to sterilize the machine itself. So while it is helpful for something like modern contexts, with ancient ones, you have to be really, really careful of contamination. And so I would not suggest doing any cryomilling. So I did a standard extraction method with a digestion. Basically, you are releasing the DNA that is trapped within all of the cells that are in the bones, and releasing it into solution so that you can condense it and collect it on a silica membrane based on its biochemistry. And so that was the pretty standard method that I used in order to basically, how does these standard methods that are the most cost effective work with these products, or work with these bone products. And there we go. So I also did, because I thought, I hypothesized that digestion needed to be a bit more helpful for the contamination issue. The skeletons were either a part of, most of them are newly excavated, maybe 5 or 6 years, or I was the one excavating them. But from excavation until lab, there's a lot of things that can happen, and a lot of contamination that can be introduced to elements, bone elements, whether it is a petrosal bone or elsewhere. So handling the individuals in any sort of way, I wanted to treat the bone powder with bleach or with a mild pre-digestion in order to help mitigate some of that contamination that I was hypothesizing was there. And so we also trialed a bleach method where you do actually put the bone powder in bleach, a very, very mild bleach, for about 15 minutes, and then wash, and then subject it to a full digestion method. There's also a pre-digestion where you add a little bit of the same digestion buffer just to kind of treat the outside of the pieces of bone, the fragments of bone, in order to, again, kind of take off that outer side. And you take that away, spin it down, and then you digest it again without including that pre-digestion, which was supposed to take care of some of the contamination. Because the whole goal is to boost your endogenous content, boost the amount of actual ancient DNA from the individual that you're working with, as well as mitigate that contamination to make sure that the modern DNA is not being introduced anywhere in the process. So the bleach method was used just on a few individuals that I thought were specifically maybe really needed it. And then the mild digestions was also trialed for a few. And all of these were compared to see what is working best. Ooh. So we also did an enrichment step. Basically, this is settled into enrichment has to be done. There's just not enough DNA that is there from African individuals, specifically because there are all these issues with how much is actually preserved. Enrichment step has got to be done. Any sort of shotgun sequencing where you're just kind of seeing what's there is not going to give you enough that's worth, it's not worth the trouble. It's not worth processing the tissue, and it doesn't give you genomes that are then valuable for you as colleagues to be useful. So I think enrichment step has to be done for any individual, for any project that moves forward. So these were done specifically for mitochondrial DNA, just again, for me to see what was viable, and just a selection of those libraries or those samples that were turned into libraries for sequencing were used because they were showing us all the signals that they were definitely ancient. There was a little bit of DNA there, and they were promising. They were hitting these marks that said it was well worth it to do that. So we used a bioinformatic pipeline, basically using computers in order to assemble genomes, consensus sequencing from all of these pieces of DNA that we collect from the sequencing, as well as fishing out or sending out all of those contaminating fungi, archaea, or any sort of bacteria that's not needed. So future work would focus on genome studies for my individuals, but for now, I have just gotten as far as looking at the mitochondrial genomes and getting those ready for analysis. So the final, the 6 individuals that made it to the deadline or for the finish line were 2 individuals from Gazali, 1 individual from the Berber Meroitic Cemetery, 2 individuals from Kiwaka, and 1 individual from Tinga. So it spanned the entire, anywhere from Napatan all the way into classic Christian period. And these individuals, there was 3 from Petrus and 3 from Tooth. And these were molecular sexed as well as confirmed with any sort of contextual data or bioarchaeological methods to confirm the individual. So we have sex estimation, but then molecular sexing. And so for some of these individuals, like the Berber Meroitic individual number 6 was a subadult. And this individual was able to be sexed with the molecular markers, even though it couldn't be with the bioarchaeological ones. And so we see there was 4 of these cemeteries were successful out of the 8 that were there. And it spanned all the way from Napatan to Christian. So it shows that there is viability in a lot of these contexts. Specifically, I didn't have, this will be in our paper, specifically what we found. While these were the 6 that made it to the finish line, meaning I have full mitogenomes for these individuals, we did see a trend where teeth were outperforming petrosal bones. This could have been in that there was another factor to it, and that was depositional environment. So individuals that are in niches that are open air, that have, like those that are found at Tengazi and Zuma and Deti, those open environments and humidity and the water table fluctuation certainly had an impact. And there was no viable genetic data from those individuals that I had access to, at least from a standard method where it is the most cost effective, but there are more advanced methods that are a little bit more sensitive, that cost a little bit more money. But it should, of course, be well worth it to invest in that. The Ghazali individuals performed beautifully. The preservation was excellent, especially when you're looking at this nice yellow dentine. This is a really good sign of preservation, at least from what I had seen with the individuals that I had access to. That really yellow dentine with minimal weathering on the outside shows that there was a good preservation of teeth under dentine underneath there. And the TARP individual, the Tinga and the Kiwaka also had these non-air niches or burials or boxed graves like those that are found at Ghazali. So the individuals that have these open air tend to maybe do not do so well, especially if they're very close to the Nile. So there's definitely a depositional environment that is impacting the preservation of DNA on these individuals. And although it was small in scope, there are some clear trends that in the upper Nubian area, teeth are outperforming Petros bones, and the depositional environment is a factor in understanding. We couldn't decide if it was closer or further away from the Nile, because also that would be a big leap to talk about, because we don't, we're not really sure. I'm not really sure where the actual bank of the Nile was and how that has been influenced in thousands and hundreds of years, but we couldn't find an association statistically if distance from the Nile would indicate that DNA could be preserved from the tissues that are there. So the final results here that we're looking at, here are some damage patterns. We're looking for certain criteria to show that these are individuals that have molecules that are acting damaged and have clear signs of being ancient. This means that they have these lesions on the tail ends of their sequences. And we look for this smile profile, where we see the lesions accumulate on the ends. So you can see that the Ghazali ones have have extensive damage. Actually, we're up here in the 25% range, and that's going 3 or 4 base pairs deep. And same with the Kiwaka. This is at 45% damage, but they still performed really beautifully. Here's the Berber individuals, a little bit lower, but again, this is a lot of damage. This is a lot of damage, but it is a criterion for saying that these are ancient individuals. So the final result, what we're looking for is a full mitogenome that has lots of coverage. So this is the human genome. Don't worry about the numbers. These are just the genes that are within the mitochondrium. This is a coverage plot to show where all the pieces of DNA could be realigned onto Kiwaka 2, this individual, and it shows really deep coverage. So good amount, so we can be very confident when we're calling these consensus sequences of what we get at the end. I'm not going to talk about DNA or mitochondrial DNA. Let's just get into the results. So we saw that we had 2 Ghazali individuals that performed well. One had very high coverage, had no missed base pairs. And this was an individual that had Eurasian ancestry. H2A is one that is a little bit high, or it's in the more northern part of, and of course, of Europe. And these are modern frequencies of heat maps of where it's more common. So you have to take yourself back a little bit to think about what it might have looked like back then. The Berber individual, T1, again, this is an individual that had a Eurasian ancestry. Same with this Kiwaka 2, also had Eurasian ancestry from the maternal side. And then we have the individuals like the L2A, another Ghazali individual, and the Tinga individual had these African ancestry signals that were coming from the maternal end. So just to show that these all have really good coverage, we look for 10 or more X coverage, so that means every base pair is covered by 10 sequences or more. For this Ghazali 5, obviously my favorite because it had 450 coverage. That is insanely deep, with less than 3% contamination. And then we can be very firm in what we're saying, that we're very confident that this is the ancient mitogenome that we're looking at. So how do these compare? A colleague of mine, Chiara Barrini, she helped me with this map analysis to understand of the individuals that are either ancient or modern that have an L2A call for their mitogenome. What do the 50 that are closely related, meaning they have less base pairs that are different, what does that mean? How do they compare to individuals? And so all of these are going to be different. All of these groups are going to be different based on the different haplogroups, but we can get an insight into relatedness to a point. It's not as helpful when it comes to nuclear variation. That's something that's also really helpful to understand ancestry. This is just maternal ancestry, and how do they relate with other individuals that are that have this same haplogroup and are very closely related. So all of these individuals have the L2A plus 143 variation. L2A is an African ancestry haplogroup, and you can see that there's a lot of representation of African, but also some Middle Eastern and East African. So some of these from Mary, from yours, are in here, I believe, and it would have been designated as ancient Kenyan. And so you would find that individual where they would be. And what we're looking at for a map analysis, these are all to scale of how different they are from that node. So that entire arm is designating that there is a lot of differentiation between the Gitsali individual and the next closest individual, which would be a West African. And so moving on to Gitsali 5, the H2A, the individual that had Eurasian ancestry, it's somewhere between the Gallian. These individuals, so this is a group, some of this is non-published DNA, mitogenome DNA that were derived from, colleagues went and they asked, have you been here for 3 generations or more? And so they were very careful about assigning these group names. And so this is a very valuable data set that my colleagues were working with and gave me access to in order to do these map analyses. So we see that this Christian individual, although it is a Eurasian ancestry, is very more closely related to these Gallian and the Mahas, which have more Nubian ancestry, where the Gallian is more Arabic. So these ethnic groups give us some insight into the differentiation but also the very, very close relatedness, even from ancient individuals, even when they spanned hundreds of years in difference. So the Berber individual was very different. This Meroitic Nubian was very different from the other ancient Egyptians, but was still the most related. So this individual goes, snaps to this magenta, which still a lot of differentiation, but it is most closely related to the ancient Egyptians, and that would be from the Schunemann paper. Same with the Tinga, an African individual of African ancestry, has the closest individual, but still quite far, would have been North African ancestry, which kind of which tracks with the L2A, because it does have frequencies in North Africa. So, and then the last one, Kiwaka 2, the H2A individual, is situated between these Gallian Mahas and the Northern Europeans. So again, these are individuals that have some relatedness. They have very similar variation. So we can see that this individual might have a little bit more to say, but all of this has to be situated within the context of Kiwaka. Same with this individual here. If you see a split node like that, it means they have the exact same mitogenome. So this individual shared it with another individual that is a very, very light purple, which might be the Egyptian. It's kind of hard to see. I'd have to double check that. But if you see a split node like that, it means they shared the exact mitogenome to show that. And this one is pretty interesting. This N1A is one of the out of Africa mito groups, and is the deepest in time, one of the deeper ones in time. So this is the work that I had been doing. A lot of that time was, does this even work? How do we get to the finish line to get these mitogenomes? The goal is to do something where we can do more population-wide comparisons to understand this, something like this, where we can see these differentiations between African ancestry and Eurasian ancestry in the cooler colors. This is this is just one that I did for haplotype frequencies from a lot of different papers, just combining everything. This is the goal, that we can look at many more mitogenomes, bring them together, and really understand what categorizes Nubian ancestry, and can we even do it? Is it so complex that maybe we can't do it? What could be very, very possible, because there is so much variation within the diversity of Sudan. It's one of the most that you see in Africa, in this East African region. There's a lot to be told. And so I'm not going to talk too much about this, but there's just an interesting story to tell, because there's evidence of gene flow outside of Africa. We have many different migration events to consider. Not only the linguistic data doesn't always match the genetic data of who is closer related to genetic affinity wise, but basically it is geographic. And there's higher genetic diversity than other ethnic groups in the Nubians than those even within Sudan. So just moving on from this, I'd like to say a lot of my work was in methodology. Does it work? Is it worth consuming the material in order to get what we get to? And I think I'd happily answer any questions about it, and we will be publishing the work that we had done to get that information out there, to understand where we should be avoiding or places we should be focusing on and why, and making sure that our stakeholders are really included within this, because it is a destructive process still. And I hope that at least some of this can get some of the information out of, should we even be doing paleogenomic work, and because it is very expensive, and is it worth it? Of course it is worth it, but not all skeletons need to be sampled because it might not just be viable, and we need to keep those for later analyses. So that's about all I have. Thank you.
Okay. I don't actually know what's going on here myself.
Stop sharing? Yeah.
Oh, that could be the reason, maybe. In any case, thank you for the presentation. That was wonderful. Thanks for that, Abby. It was very informative. I still can't really see anyone, but I'm assuming this is going on track, so I will progress as per usual. So thanks again for the presentation. We'll move to the final presentation for today, which is being given to us by Dr. Mary Prendergast. Dr. Mary Prendergast is an associate professor of anthropology at Rice University. Dr. Prendergast's research focuses on the interconnectivity of human and animal worlds, addressing questions around pastoralism and how foodways were transformed through interactions amongst people who had diverse food gathering strategies, problematizing labels such as herder or forager, with a primary focus on areas in Eastern Africa, in particular in Kenya and Tanzania, where Dr. Prendergast co-leads excavations at the pastoral Neolithic site of Luxmanda as part of a collaborative project with the National Museum of Tanzania and the University of Florida. So thank you for joining us today, and Dr. Prendergast, the floor is yours.
Thank you very much. I'm going to assume that you guys can hear me okay, and that you can also see the slideshow that I just started sharing. So if there's any trouble, just let me know. I am going to sort of shift our focus southward today, substantially southward. I do not work in Nubia, as Robert just pointed out, but I hope that some of what I present today will be relevant to those of you who do. You've already seen this, and both Kendra and Abagail have given a great overview of the very rapid rise of ancient DNA studies worldwide, the extent to which Africa has played a role in ancient DNA analysis, as it's already been shown, extremely minor role. So I think I can move through this pretty quickly. The one thing I'll say is that even though, as Kendra noted earlier, African genomes contribute only 3% of global ancient DNA data right now, genomic data, from an Africanist perspective, right, as somebody who works on the continent, works in the Holocene, it has actually felt very rapid and overwhelming at times. We went from only 2 papers as of the year 2015 that were really pretty pioneering in African archaeogenetics to a really overwhelming number of papers. I'm sure I don't have every single one of them here, but one thing I wanted to just highlight for those of you who are relatively new to this discipline is there's a few good reviews out there. And I'm not just saying this to try to pump our own work, but there are just not that many reviews. So 2 of them are through the Oxford Research Encyclopedias. Those are both open access. I'm pretty sure that Mario Vicente and Karina Schlebusch's article is also openly accessible. But these are good starting points if you want to just get an overview of what has been happening across the continent, what problems there are, and what the future holds. In the review that Kendra, Elizabeth Sawchuk, and I wrote together, we summarized the state of the field such as it was when this paper was written in 2021. And as has already been pointed out by both Kendra and Abagail, there's tremendous patchiness across the continent, both in terms of the spatial distribution of individuals whose genomes have been sequenced and in the chronological distribution of those individuals, right? So you can see these major gaps that have already been highlighted, huge geographic gaps, obviously, as well as chronological gaps. And this paper does not reflect a paper that we published, led by Mark Lipson last year, that has increased the number of ancient genomes from the late Pleistocene in sub-Saharan Africa. So that's very exciting. But nevertheless, most individuals that we study date to the last 2,000 years, are relatively recent. Abagail and Kendra have already highlighted a lot of the reasons for these gaps. Of course, ancient DNA preservation is a huge part of this. There are lots of other biases too. Certainly the accessibility of collections in many countries remains an issue. I think that historically geneticists were interested in other kinds of questions rather than those that preoccupy those of us who work in Holocene Africa. But also these biases do to some extent reflect real archaeological research biases. And something I'll say today, and that I say every time I talk to a geneticist audience actually, is, if genetics researchers want to do this work, then either directly or indirectly, they also need to support archaeological and curatorial work on the continent, because Holocene African archaeology remains grossly underfunded, particularly south of the Sahara, relative to other world regions and relative to other time periods. For example, human origins research south of the Sahara is comparatively quite well funded. So I'm going to hone in today on one particular case study, and actually one particular paper that I was hoping might be relevant to those of you who work in Nubia. But I'll just say this paper is part of kind of my broader interest as an archaeologist and a zooarchaeologist, right? My training is really in faunal remains. I'm not a bioarchaeologist, I'm not a geneticist. And my long-term research interest is in understanding African pastoralism. And so for those of you who know this literature well, there's lots of questions about the means by which domestic animals such as sheep, goat, cattle, and donkey were introduced throughout the African continent, the routes by which these animals were introduced, the chronologies of these introductions. I'm saying introduction, but I'm sure, especially those of you guys working in Nubia know there's also longstanding debate about indigenous origins of African cattle. And then, of course, there's a big debate, especially as you move into my area of research, Eastern Africa, and then even further south into Southern Africa, there's a really ruckus debate about the extent to which pastoralism spread through the movement of people or the movement of animals through, down the line exchanges, right? Movements of animals and ideas. And it's important to remember that tied up in all of this is the longstanding indigenous foraging economies of the region. And although we have a sort of dominant cattle before crops narrative in the archaeological literature, that's a reference to the work of Fiona Marshall and Lisa Hildebrand, who showed that pastoralism spread well before farming across most of the continent south of the Sahara. Increasingly, that narrative is being questioned as we start to uncover previously invisible evidence for plant use, And we're starting to reevaluate the roles of wild game and fishing, plant collection and cultivation in this spread of pastoralism, rather than seeing this as some kind of wave of livestock herders. Another question that has come up, and by the way, the questions that are on this slide are all things I think can be addressed using genetic research, right? That's why I brought them here, are whether or not we really can attribute particular material culture traditions to particular groups, the old peoples and pots question. There's a lot of investigation now into not only the spread of pastoralism, but specific genetic adaptations to this new lifestyle, particularly with respect to digesting milk. And then, of course, there's the thorny old question of the extent to which linguistic histories, increasingly what we can detect from genetic histories, and the archaeological record overlap or don't. And actually, I always think it's more interesting when we don't see nice, neat overlaps. So the papers that have come out focusing on pastoralism are threefold so far, one in 2017, one in 2019, and one in 2020. I'm going to be focusing primarily on this paper that I led and that Kendra is also a part of from 2019, which I should say by ancient DNA standards is like the Pleistocene. So there are certainly things that have come out since that have built upon this work, but I still think it's worth sort of thinking through the problems that this paper has raised for us as archaeologists. And again, if you want user-friendly things, there's a nice, very user-friendly conversation piece about this that Elizabeth Sawchuk and I wrote when the paper came out. This paper was part of a much larger project that is still ongoing, in collaboration with curators and archaeologists in 3 countries and at 3 major national museums, the National Museum of Tanzania, National Museums of Kenya, and the Livingstone Museum in Zambia. And so I just want to acknowledge all of the people who've been key parts of this project. And then, of course, as you know, there are many, many more people involved in any ancient DNA study who are not acknowledged here. And what we did in the 2019 paper is we sequenced the genomes of 41 people who had lived over the last 4,000 years and were buried at archaeological sites in present-day Kenya and Tanzania. And these individuals, the aim of the study was really to investigate early pastoralism. Inevitably, of course, when you're doing this kind of study, you wind up encountering sites where you don't have such a clear link to early pastoralism. So we also had individuals who were buried and associated with evidence for foraging, as well as much later farming and metallurgy. So we have individuals who lived during the later Stone Age, the pastoral Neolithic period, and then the Iron Age. And so the pastoral Neolithic period, of course, is when pastoralism spreads into Eastern Africa. The Iron Age is when we begin to see the earliest evidence of farming and metallurgy. And so my focus today will be on this pastoral Neolithic period. And I will just mention that the individuals whose genomes we studied were associated with 2 different cultural traditions, material traditions, which are known as, well, the pastoral, Savannah pastoral Neolithic, which is a catch-all term that's really problematic. More specifically, they tended to be associated with this Narasara type of pottery that you see on the left. Other individuals were associated with what we call Elmontitan pottery. And so for our purposes here, just know that these are 2 very different material cultural traditions, not only reflected in pottery, but also to some extent in settlement patterns, obsidian sources, lithic technology, burial patterns, right? So these are typically thought of as, by Africanist archaeologists, as 2 distinct cultural identities. And some linguistic historians have even posited these represent very different so-called ethno-linguistic groups who would have, whose ancestors would have come into Eastern Africa at different times. So we're going to just spend some time looking at the results of this study in a PCA, right? You've thankfully already seen a bunch of PCAs, so I don't have to explain how these work. And what you see here is a large number of present day groups. Okay, so those are in circles, small circles. And then the ancient individuals, both from our study and from what had been published at that point when we were working on this paper in 2019, which was very limited compared to what's been done now. that are then placed into the PCA along with the present day individuals. And I'll just point out two things of interest for us. The green dots represent Northeast African groups. There's a line of relatedness among people with, let's just say very broadly, Northeast African ancestry, stretching from people who identify as Copts up here on the upper right-hand side, who are in close position to individuals from Southwestern Asia, including ancient individuals from Southwestern Asia. This is that Israel Chalcolithic group here, all the way down to Dinka and Nuer at the more extreme position in the lower left hand. The purple dots represent, the purple dots represent Afroasiatic speakers. I don't know why it's disobeying me, who live today in Tanzania, Kenya, and Ethiopia. So again, this is present day data. We all know perfectly well people move around and have very complex histories. So we have to sort of, that's why we have the ancient DNA record, right? Is to try to sort of ground truth some of the inferences that we make based on present day genetic and linguistic distributions. But what you see here basically is these two kinds of relatedness among these present day groups, right? What we called Sudan related group or Northeast African group in green, and the Afroasiatic speakers of Eastern Africa in purple. And the vast majority of the individuals in our study and all of the individuals who are associated with early pastoralism cluster here at the sort of nexus of these two kinds. But a PCA can only tell you so much. When we started to go into more quantitative analysis, we found that they were best modeled as being more closely related to present day Northeast Africans, especially Northeast, Sudanese. So let me just point out a couple other things here, and then I'm gonna try to summarize what this all means. So a couple of very interesting individuals in our study that I'll hopefully have time to circle back to at the end. As you can see, are outliers here, these two individuals. These two individuals were a real surprise to us. They were buried at a site that had never really been documented properly, and I'll try to talk about that at the end of my talk, called Pretjohn's Gully in Kenya. They, to our surprise, both were directly dated to 4000 BP, which is extremely early for any sign of pastoralism at this point in this part of the Rift Valley. And they do not have associated archaeological record. So I always like to sort of take this with a grain of salt, because obviously genes don't tell you what people ate or whether or not they owned livestock, right? The reason we're calling them possible early pastoralists is because of their relatively close proximity to other individuals associated with archaeological evidence of pastoralism, who are indicated by the other symbols that you see here. So these sort of yellowish squares and circles, the red circles, right? These are individuals we associate with pastoralism in Eastern Africa, ancient individuals. So those two individuals at Pretjohn's Gully, I'll try to come back to later. Another thing that we noticed, again, thinking about these differences between Elmantite and the Rossera pottery, is that there was absolutely no genetic differentiation between individuals buried with these two material cultural traditions. And again, this is not only reflected in the PCA, but in a series of statistical tests that we then performed on these individuals, which I will not get into in any detail. And so, you know, that to me as an archaeologist raises all sorts of really interesting questions about these folks living basically side by side at, as far as we can tell from the radiocarbon record, the same time, who are doing things really differently, yet clearly shared many recent ancestors. And that's a really archaeologically and culturally interesting problem that I think merits further investigation. Other things worthy of note are that once we get into a later time frame called the Pastoral Iron Age, so this is about 1,000 to 1,500 years after the spread of pastoralism throughout Kenya and Tanzania, we then get a much later influx of iron-using herders who many present day Nilotic herding groups in Kenya and Tanzania trace their ancestors through oral history to these groups. They come in, and as you can see, the 4 individuals that whose genomes we sequenced, who date to this later Iron Age period, all of them date to about 1200 to 1000 BP. Have a real, they're very shifted in the PCA towards this end. If you recall, this is where the Dinka and Nur sit, present day Dinka and Nur. And that really resonates with oral histories that are maintained today of these pastoral Iron Age populations and represents an influx of Sudan-related ancestry. And then finally, I will not talk about this today just because it's outside of the scope of our interest here. But we had a single individual from a site called Delarine Farm. For those of us who work in East Africa, this is a really well-known site because it's the only site that we have with early evidence, direct botanical evidence of farming in the Central Rift Valley. And that individual, as you can see, is shifted very close to West African individuals from Nigeria and Sierra Leone, right? Again, present day individuals. And that, of course, resonates with the idea that farming in Eastern Africa is associated with the spreads of both West African related ancestry and Bantu languages throughout the rest of Southern Africa. So this diagram I know can be overwhelming, but this is kind of a summary of those results. And what we concluded at the end of the study was that we had documented a sort of 4-step spread of foodways, different new foodways, right, beyond foraging into Eastern Africa. And I'll try to make some sense of this diagram. For our purposes, I think the most interesting thing to think about is here. We know that there are basically 3 major components of ancestry in the individuals that form the heart of our study. Those 2 early pastoralist individuals that date to 4000 BP at Pretjohn's Gully, the sort of mystery site, and then all of these individuals who are associated with the pastoral Neolithic, with evidence of pastoralism in Kenya and Tanzania. So just thinking about those 2 groups of ancient individuals, they can trace their ancestry to 3 major sources. One source, of course, is indigenous foragers in Eastern Africa, and that's not a big surprise. We would expect that no matter the strength of cultural boundaries, there may be at least some admixture with indigenous foraging groups whose ancestors had lived in the region for thousands and thousands of years. Then we have these other 2 groups. The proxy that we used for these recent Sudanese groups was the Dinka, right? We were using the Dinka as a reference group. We are not saying the Dinka are ancestral to these individuals, but rather that in our models, they are the closest related individuals to a putative ancestral source, right? For which we do not have ancient DNA data. Same here. We know that there's another component, which is a component that actually both Kendra and Abagail talked about, this West Eurasian ancestry. We use here as a proxy that Israel Chalcolithic group of ancient individuals. And again, we're not saying that the herders of Eastern Africa are descended from those Israeli Chalcolithic individuals, but that was the group available to us at the time in 2019 that was most closely related to the ancestral source. What we really don't understand from an archaeological or genetic perspective is how these different groups come together to ultimately form the sort of tripartite mixture of genetic ancestry that is the basis for the pastoral Neolithic. Nor do we have a great record, archaeological record, although we do have some oral historical records, for how that influx of increased Sudanese ancestry comes in the Iron Age. So from an archaeological perspective, what I wanted to spend the 5 or a few more minutes that I have left thinking about is what are the questions for us as archaeologists curious about tracing the spread of pastoralism into Eastern Africa, where relatively speaking, it's quite well documented, right? We have lots of early pastoralist sites in Kenya and Tanzania that have been the subject of decades of study. And the question really comes because we do not have any relevant ancient DNA data yet, right? Kendra and Abigail have both presented a lot of wonderful data that is dating to after the time frame of interest here. What we really need to do is kind of target archaeological sites and individuals in this time frame in which we have estimated that admixture amongst these components took place. And that would have occurred for the first time around 6,000 BP, continuing on until about 4,000 BP. That's based on some admixture date modeling that we have done with pairs of individuals in our samples. And so just to sort of zoom out for a second, of course, that 6,000 to 4,000 BP window is at the tail end of the African human period, marks a tremendous moment of change for people who had been living along the lakes and rivers of North Africa, had been herding cattle, fishing, hunting, collecting wild plants, right, cultivating plants. And ultimately, the drying conditions of the end of the African humid period are widely thought to have been what prompted herders to move southwards into Eastern Africa. And actually, you can just barely see here Lake Turkana, where we have the earliest evidence of herding in my region of focus. And I just wanted to point out, I just pulled this slide in this morning, a really nice paper that came out. I saw it for the first time yesterday, that is a wide-ranging review of zooarchaeological literature, as well as organic residue analysis on pottery from a number of key sites across North Africa. And one of the points that's driven home in this paper is that there's not sort of a wave of advanced model for pastoralism. It's really quite sporadic. People continue to fish and hunt, perhaps exactly as we would expect. I mean, I don't know, working in East Africa, that's exactly what we expect. And that's what we often see, in fact, even ethnographically in some cases. And I think that's important for considering the sort of social and economic context in which admixture may have occurred in Northeastern Africa and expansions may have begun. So what are the kind of key problems and challenges that we face here? Again, I know I'm speaking to an audience of Nubianists, right, and of archaeologists. Well, the archaeology of early pastoralism is not very well documented once you move south of Nubia. South Sudan and highland Ethiopia in particular are incredibly interesting. And for many reasons that are geopolitical and financial and other, have not been very well investigated in terms of early pastoralism, in terms of this key time frame. There are some sites, there's of course, well-known so-called cattle culture sites in Northeastern Africa, right? I mean, Nabta Playa is probably the best example, but all of these incredible sites that some archaeologists have tried to connect to the monumental cemeteries, these really elaborate cemeteries in the Turkana Basin. But those connections are very tenuous at best. And we truly don't understand how the first herders in Eastern Africa, who have this amazing archaeological sort of material culture tradition, emerged. We don't know anything about them. And unfortunately, despite our best efforts to sample extensively in Lake Turkana, we have not been able to recover any ancient DNA. And that brings me to the next point. As Abigail and Kendra both pointed out, there are huge challenges in ancient DNA preservation in Africa in general, and perhaps especially in the parts we're most interested in. Something that's really been lacking are the kind of meta studies that would help us understand better what optimal preservation conditions are like, right? We have some sense, rock shelters, caves, high altitude places, right? So the Ethiopian highlands, for example, maybe even amongst individuals, right? We've noted that juvenile individuals tend to preserve DNA better than older individuals, but really that work still remains to be done. Something that I personally would really love to see done, especially because it would mean less destructive work on human remains, is using faunal remains or sedimentary ancient DNA as proxies for understanding human cultural dynamics. And of course, that work is happening, is underway, both faunal remains and sediment work, but we're still awaiting a lot of results. And then finally, of course, there's lots of other archaeological proxies you can get into if you're interested in the spread of herding. Again, a lot of work's been done on organic residues and on, to some extent, lactase persistence. but more needs to be done. And finally, the last thing I'll say is that there's huge challenges, and this faces all of us working in African archaeology, in sampling legacy collections. I'm gonna, because I know I don't have a lot of time, I'm just gonna illustrate a couple of these points with a few slides. One thing I would just love to say is, even though this slide is from 2018, which again, in ancient DNA terms, that's like the Pliocene, really hasn't changed with respect to African faunal and botanical ancient DNA. We have a handful of studies from Southern Africa on livestock, but that's about it. There are key reasons why faunal ancient DNA studies haven't worked, but I do think that that's the area we really have to focus our efforts. I'm gonna skip the next slide actually, because I know I don't have a ton of time, and just focus for one last moment on the challenges of working in legacy collections, which I know anybody who works in Nubia also faces. A huge part of the challenge we encountered in this paper was tied to both provenience and provenance. So figuring out where contextually things came from in sites was often a challenge. We had things like this, which was in one case, the best documentation of provenience we had. And then there's problems of provenance. There has been a tremendous amount of movement of archaeological material, including human remains, for example, across the former British Empire. So we have materials from Kenya and Tanzania that are in Zambia or in the United Kingdom. A lot of material from Tanzania was taken to Germany when Tanzania was still a German colony. So this raises, of course, all sorts of key issues around repatriation, as well as permissions and access. And then, of course, there's the problem that we often lack archaeological records. So I'll just kind of close with this by saying that the two individuals I mentioned earlier from this apparently early pastoralist context do not have an associated archaeology. They were associated with this geological trench, an environmental study, not an archaeological study. And just last week, I came back from Kenya, where we spent a week hiking up and down the walls of this gully, trying to find the site again so that we could re-excavate, better understand the chronology, better understand the cultural history of these two people who right now are genomes without an archaeology, in order to both provide them with a greater sense of personhood and give something back to the museum, who initially asked us to do this sampling because they knew nothing about these two trays that were in their collection. And that's the kind of stuff I think that we all need to be doing, right, if we want to do the ancient DNA work. We also really need to invest in the attendant archaeology and curation. So I will leave it there so that I know we're already at the end of time, so that we have sufficient time for questions. Thank you so much.